In the IP-FCM experiment, protein complexes are captured on beads in order to identify and study the different proteins interacting together. First, an immunoprecipitating antibody specific for a protein of interest is covalently coupled to polystyrene latex beads. As a second step, these beads are incubated with cell lysates, allowing the specific protein and interacting proteins to bind to the beads. The beads, together with the captured proteins, are then washed and probed with fluorescently labeled antibodies specific for various molecules that may be associated with the immunoprecipitated protein. Finally, the fluorescently labeled beads are analyzed by flow cytometry to determine which proteins are present and which are absent from the captured complexes.
The advantages of this technique over other methods, such as immunoprecipitation followed by Western blotting, is the ability of IP-FCM to allow assessment of native protein-protein interactions without requiring genetic engineering or large sample sizes. IP-FCM can yield a high signal-to-noise ratio even when very little biomaterial is available for analysis, as it can be performed with small volumes and low concentrations of analytes.
To begin this procedure, prepare the following buffers: MES coupling buffer, quenching blocking storage buffer, abbreviated QBS, and flow cytometry or FCM buffer. Determine the concentration of the carboxylate modified polystyrene latex beads or CML beads by diluting in PBS and counting with a hemocytometer. Once the concentration of the beads is